Welcome to the first Epilepsy Grand Round of the George Washington University 2013 Epilepsy Grand Round Series. For our inaugural talk this morning, I'm delighted to introduce Professor Dominique Durand from Case Western Reserve University. Dr. Durand is the E.L. Linseth Professor of Biomedical Engineering, Neurosciences, Biophysics, and Physiology, and Director of the Neural Engineering Lab at Case. Dominique earned his engineering degree from Toulouse in France and his master's in biomedical engineering from Case Western and his PhD in electrical engineering from the University of Toronto. He has earned numerous awards, including the NSF Young Investigator Presidential Award, and has mentored a big number of researchers who have gone on to become prominent figures in biomedical engineering and neuroscience. His research has focused, among other areas, on the interaction of electric and magnetic stimuli with neural tissue, both in the central nervous system, such as for the treatment of epilepsy, and the peripheral nervous system. And he has earned funding from the National Science Foundation, National Institutes of Health, and other organizations over the years for his research. He has published over 100 papers in major journals, and is the editor-in-chief and founding editor of the Journal of Neural Engineering. Without further ado, Professor Durand.
Well, thank you very much, Mohammed. It's a pleasure to be here. I have the honor to be the first speaker in that series. It's got a special honor. And so today what I will talk about, as asked by Mohammed, is some of the work that we've been doing on the interaction between electrical stimulation and epilepsy. As you heard, I'm by training an electrical engineer, and that's still my love, but I'm very also in love with neuroscience, and particularly the interface. How do these two fields interact? And in this particular case, how do electric fields applied to neural tissue can control it in some extent? So I remember working on this area a long time ago. In fact, my PhD was on a different area, but my first experiment was on epilepsy and electrical stimulation, and at that time was interested in how a single pulse, as in cardiac tissue, can reset the activity of neural tissue. And this colleague of mine told me, well, what are you talking about? If you stimulate neural tissue, you're going to excite. How are you ever going to stop seizures that way? That doesn't make any sense. But I pursued this idea, and it's been a long, long journey. Over 30 years of work trying to understand how electrical stimulation can suppress seizures. So what I'll do is I'll give you a little bit of a history of electrical stimulation, and a little bit of fundamentals, only a few slides of how electrical stimulation works. And I will take you through a broad spectrum of the effect of electrical stimulation on neural tissue from a frequency point of view. And I'll show you some results that we've been able to get. So here's it's one of the very first stimulator electric fish, which was applied to patients for curing everything, including gout, a long time ago, even as far back as the Greeks were concerned, with obviously no understanding of what could happen. But this fish can generate several hundred volts of electricity and can definitely do something to the body. Whether it has ever been used therapeutically with success, that's a different story. But for a long time, people thought that electricity and biology did mix and could bring some therapeutic effects. Here's the real first experiment to understand a little bit about how electricity and neural tissue are connected. This is an experiment by Galvani in 1790. And he put this piece of metal between the spinal cord there and the leg, and you can see the leg is moving. And he did not know what was happening. This is zinc and copper here. He thought there was something passing between this point through the wire into the leg that made the leg move, called animal electricity. Turns out that this was solved by one of his contemporary, Volta, who realized that the combination of zinc and copper can produce an EMF, electromotive force, that can then generate electricity and activate the nerve and move the leg. So that is the very first interesting experiment. But here's another one, not that much later. This is Benjamin Franklin, and he is working with this machine, which is recognizable because it's clearly an electrostatic machine. You can see the crank here. You turn this by friction, you build up charge. This charge is stored in this jar here. This is called a Leyden jar. This is basically a capacitor. And when the capacitor is charged, you can then discharge it. And in this particular case, you see that there will be a spark between these 2 points here. This happens to be a woman who's paralyzed. So already in 1799, it was possible to demonstrate that electricity could actually move a limb. And I like to point out the difference between what's going on here and what's going on now in terms of electricity for restoring function in patients. I hate to say we haven't made a huge amount of progress. And I'm as guilty as anybody else of that because I do work on this field as well. And there is some progress has been made, but clearly there's a long, long way to go. And if I had time, I would tell you what the reasons are, and I have some good ideas what those are. But so you can see right there what was an experiment that was actually quite useful in figuring out the interaction between muscle tissue and electricity. This is an experiment from Paris. This is sort of an interesting experiment because you can see that this patient is between a very large charge, a negative charge generated by this electrostatic machine, and the ground plane is here. And this is called a negative ion douche, where the patient is exposed to this. These negative ion machines are still popular in Europe because they clean the air and they're supposed to have good benefits. But here in the US, it's not being approved by the FDA. So you don't have any device like this for therapeutic purposes in the US. Now this is a device that made a huge improvement. That is a machine that is a stimulator based on Faradaic principle. So we switch from electrostatic to Faradaic, and Faraday obviously showed that you could generate electricity by induction. So you can see that stimulator is much smaller than the electrostatic machine. In Cleveland, there is a museum that has this machine in it, and sometimes when I teach the class, I go and pick one up and show them to the students. You hold these two handles in your hands there, and you crank the machine, and you can get a jolt of electricity. I'm not sure what this patient was doing. It looks like pain in the arm. So just give you an idea what's going on. At this stage, still very little is understood about how electrical stimulation affects the nervous system, even though these machines were found in most physicians early in the 19th century. So let me show you 3 slides of how electrical stimulation affects axons and cells. This is a very brief summary, but it gives you an idea of what's happening. So take an axon, a mallet in this case, and put an electrode right here, an anode. In this case, there will be a plus source here. And if we pass current into this position, the current will flow radially in all directions. Some of that current will enter the axon and produce action potentials that will flow in 2 directions, this direction here and this direction, so antidromic and orthodromic. It's very easy to calculate the voltage along the axon. Here's the formula right there. You can see decays as 1/R. And here's a plot of this voltage along the axon. It's maximum under there. Give you some idea of what's happening if you inject about a milliamp at a distance of about 1 centimeter, the resistivity of tissue is about 100 ohm centimeter, you get about 80 millivolts. So it's a significant number What matters, of course, is the transmembrane voltage that needs to be calculated. In some cases, people use dipoles, particularly if you think about those DBS electrodes are in brains. You put currents plus and minus in two adjacent electrodes. In that case, the voltage is very different. Here's the current here, one plus, the minus, and you can see the voltage profile. It has to be negative under this electrode and positive under this electrode, so it looks like this. You also notice that I use the same current and that the voltages are much, much smaller. The equation is given by here, and you can see that the voltage decays a lot faster, 1 over r squared. So if you use a dipole, clearly the voltage will, you will need a lot more current. And the reason is that this current here that you apply is multiplied by this distance between the two, and usually this is pretty small. So then this product is small. So that means that dipoles are good at confining the electric field close to their location, but they also need a lot more current than the monopole. And then finally, I just want to bring out one important point that says a lot about electrical stimulation. Because what really matters is the voltage across the membrane, and because that's where the sodium channels are. And this voltage here is actually given by this differential equation, second order. I won't bug you with the details, but what drives this equation, the source is this term right there. So if this term is 0, nothing happens. But if this term exists, then we have excitation. And this actually is the voltage outside the axon. So what you can see from this equation, it's not the voltage that excites. It's not the first order derivative of the voltage. It's the second order derivative of the voltage that excites an axon. So if you have a non-zero second order derivative, then you will be exciting the nerve. And this is important because it explains one finding that cathodes are actually better at exciting than anodes. Excitation near a cathode will be better than excitation near an anode, and you can see why. For the same current where everything is reversed, the cathode will produce a voltage like this, and the anode will produce a voltage like that. You see these two are symmetrical, but because the depolarization for axon is much larger for an axon than this depolarization here, right, then this cathode will be a lot better. So there's a lot of understanding now we have about the principles of electrical stimulation and how an electrode here can activate axons and cells. These are very simple principles, but important to understand it. So now we can go into the topic of the day, which is how do we control neuronal excitability with electrical stimulation. And there's a lot of confusion about this topic because there are so many different frequencies that are being used for doing this. So I've decided to break it down into different ranges which are known in the field. So I will start to tell you a little bit about what's going on with very low frequency, DC. Amazingly, DC frequency can do something very interesting. Then there's a very low frequency, which in the range of 1 to 10 hertz. There is another range of frequency called intermediate between about 20 to 40. I won't talk about this, although this is the frequency range that's used for vagal nerve stimulations. There is another very important range called high frequency, from 50 to 400 hertz. That is the range of frequencies used for controlling motor disorders, as in DBS, deep brain stimulation of Parkinson. And I have some data to show you on this. And then finally, there's another range of frequency, very high frequency, 5 kilohertz to 10 kilohertz, even up to 30 kilohertz. This is sort of a fairly new area that is used currently only in the peripheral nervous system for controlling pain. But still very interesting because you can block actually axons using this technique. I will not talk about this. So I will talk about the very DC, this range, and this range. So here's what happens with very low frequency DC. Something interesting. This is actually quite old experiments that we did a long time ago. We use to do these experiments what's called the hippocampal slice, which is a slice, about 400 micron slice of the hippocampus. In the rat, the hippocampus is very differently located than in humans, but it's a banana-like structure ranging from septal to temporal. And if you cut it transversely, then you end up with this structure here, which contains the CA1 cells, the CA3 cells, the granule cells, and it contains all the connections to these neurons intact, more or less. So it retains this structure, and so it's very interesting to work with because you can actually, under the dish, you can see the structure right there. You can place your electrodes. It's very easy to work with. So it's very popular, very convenient for studying epilepsy. And in this particular case, we generated epilepsy with penicillin. Penicillin in high concentration causes seizures. And so we put it in this bath, and you get this sort of activity right here. You can see in response to a single pulse, instead of getting a single response, you get multiple response, meaning that the neurons fire here, 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 they're very excited. So this is not what you call a seizure, but this is a state of hyperexcitability that could eventually lead to a seizure in a slice. But if we place an electrode right here, I call blocking, right where the neurons are, and pass a small current, you can see that as with current as low as 50 microamp, this response decreases until it's completely gone. And if you turn up, you can see the plot of the percent suppression here as a function of current. As you get better and better. You get complete suppression for a fairly large range. And eventually, you get excitation here because at the onset of the pulse, then you get excitation too. So clearly, these currents can do something. And we sort of pursued that a little bit with a different model to see how robust it was. This is an interesting model of epilepsy where you remove calcium from the tissue. If you remove calcium, synapses are no longer working. Yet, you have this very strange activity here that continuously go on. This is about 1 or 2 few seconds range, and this is hyperexcitability here, and you get this baseline shift. The interesting thing about it's not clear how this is arising, and it's not even clear how it's propagating, but it is. And so it's continuous and spontaneous. In this case, what we did, we placed 2 electrodes, one here and one there. We generate an electric field, uniform across the tissue. And we were able to detect this event and apply stimulus. And you can see what happens here. Here's a normal event. As you apply 2.9 microamp, this gets smaller, smaller, disappears at about 4.4 microamp. If you reverse the polarity, it actually gets worse. And here's a cumulative histogram here of this experiment. You can see that it's possible to block everything with only 8 microamp. I mean, 8 microamp is really small. These fields I'm talking about are in the very low range, the smallest range of what is required to even affect neurons. And you can actually block 15% of the population of neurons with only 1 microamp, and that's really small. So there was some interest at that point to pursue this and see if something that could be done with it. And here's how it works. It's well understood. Here's a cell right there. Here's an anode. So if I'm passing some current at this point next to this cell, current will enter the neurons and leave in the dendrites. And if you put a cathode, the opposite will happen. The current will leave at the soma. So we know already from analysis that if current enters the membrane passively, it will cause hyperpolarization. If it leaves the membrane, it will cause depolarization. So what happens here, if the anode is located next to the soma, this whole region will be hyperpolarized, and the cells won't be able to fire. Because the current must leave, it will do that in the dendrites, but there are no sodium channels there, so it doesn't matter. So basically, this is quite effective, but there are some problems. One of them is big problem. They have to be oriented exactly in the right direction. So I have to have the field in this direction. It has to be longitudinal with the cells for this to work. And it's very hard to find any place in the brain where everything is lined up just right so that you can pass current and inhibit cells. The other big problem is, you may not know this, but when you pass DC current in the tissue, directly that causes a lot of problems because electrodes polarize and they generate changes in extracellular space, pH, superoxides that damage the tissue. So I did not pursue this, but a colleague of mine did, and I think got a grant on it. I don't think he went very far with it because there are some problems. But this technique has been resurrected in some ways. And this is now in clinical trials. This is a different way of doing DC stimulation. Instead of placing electrodes in the brain like I was doing, you place your electrodes outside the brain and getting a really good conductor, say like a sponge filled with sodium chloride, for example. You put one here, you put one on the other side, you pass current across the brain. These currents are very weak. The electric fields generated in the brain are in the range of about 4 millivolts per centimeter. This is lower than what's known to be effective, so it's not clear how it works. But there are some effects. And so there's been a lot of papers, especially here in Washington NIH. There's been a lot of work on this. Here's an example of an evoked potential. And you can see, depending on the polarity of the stimulation, you can clearly affect the amplitude of this potential. If you apply this field while somebody is doing word counts, for example, you can affect the error rate and the way of the amount of memory that's been produced. So there is some effect there, but not understood. One of my students in New York is actually very involved in this, trying to understand this technique, and did a lot of computer modeling of the brain, the currents, etc. So it's very interesting because it's non-invasive, it's outside the brain, but it's not clear, it's not localized, and the mechanisms are unknown. Still, there's some interesting potential there. Maybe even for epilepsy, although I don't think people have tried it. So now let me try to tell you a little bit about a different frequency range. So going from 0 hertz, right, to 1 hertz, big step, big step. And so here's one of the very first experiments I remember about low frequency stimulation. This was done by Yamamoto here and Tergao, using a magnetic stimulation. You apply this to the brain, pulse, boom, boom, like this. And there was a clear effect. You could see a decrease in seizures, not very significant, but definitely there. So from that point of view, it was sort of clear that LFS could, low frequency stimulation, I call it LFS, could lower excitability in the brain, and it's in the range of about 1 to 10 hertz. So we sort of pursued this in a lot more detail with a different approach, but still getting that, using that idea that low frequency stimulation can do something. So we targeted patients with mesotemporal lobe epilepsy. At that time, we were thinking about this, but we were working on animals only, doing electrical stimulation in rats. But this is a big problem. I'm not going to go into details because this audience clearly knows all about the prevalence of this disease and the fact that surgery does work, but there's a lot of patients that cannot have surgery for multiple foci surgery, so perhaps stimulation can help. So what we did is picked a target, and the target that we chose based on some of the ideas I was working on at the time. Basically, I wanted to find a target that activates a large part of the brain all at once. Very much anti or different from the current ongoing ideas that we should put a stimulus right in the focus of the stimulation. This is a theory that's been used by several companies and several studies. Let's find the focus, put an electrode there, apply frequency, a pretty high frequency stimulation, and then you can stop seizures. At that time, I knew a lot of people were doing this, and I took the opposite view, and I said, let's take a target that activates a large part of the brain all at once and prevent seizures in all this area all simultaneously. It comes from the fact that if you think about putting an electrode in the focus and detecting the focus and applying stimulation when the focus is starting to generate the seizure, my understanding of signal processing and signal detection was that it might take still a few seconds for these seizures to be detected, and by the time the algorithm detects it and applies the stimulus, the seizures might be gone. So not that it doesn't work, but it may not work all the time. So the idea, can we then instead prevent seizures from spreading everywhere? And so we tested this idea, and this is a cross-section or view of the hippocampus in the rat. There is one track called the ventral hippocampal commissure that connects the two hippocampi. These axons go right into the CA3, CA1 regions on both sides. In fact, these axons are the axons of CA3 cells on one side and cross on the other side. There is a dorsal hippocampal commissure in rats, and that dorsal one is much smaller and connects the entorhinal cortex on both sides. But we decided to pick this target because it's a lot stronger. So we could put electrodes in these targets in CA3 and record also in CA1, and while we're stimulating, and we picked a frequency of 1 hertz, and I'll show you what happened. So we developed, to test this idea, we developed a new slice preparation that just been published, like I think it came out a few months ago. I've shown you this slice preparation before with a transverse cut. This is a preparation that was actually developed by one of my students, MD PhD. She's about finished almost. And she decided that we wanted a bilateral slice. So she took the brain and cut just basic horizontal slice until she hit this commissure, preserved the commissure, so preserved the one slice that had the two hippocampi together and the commissure still connected. So that lives pretty happy in the dish, and you can put an electrode there, and you can record evoked potentials on both sides. And if you apply a drug, you can get epilepsy to happen here, and there's some interesting happens. So here's some of the seizure-like activity that we observe in this prep. It's very, very hard, in my opinion, to get real seizures. Well, there's no real seizures in vitro, but seizure-like activity in vitro. But in this prep, we were able to get some decent-looking seizures. By decent-looking, I mean a seizure that lasts more than a few seconds. Usually, you get a lot of this activity, which is I call interictal activity, although clinicians, I don't know if they'll call this, they might call this a seizure, I'm not sure. I've run into several disagreements on this. We call this interictal activity, and this sort of high-frequency activity, we call that seizures in animals. But we were able to get pretty decent seizures developing in this slice. We use a model of epilepsy called 4AP. 4AP is 4-aminopyridine, blocks a potassium channel that repolarizes the action potential, and it's highly epileptogenic. And so you put it on, and you see this activity starting, and you get a seizure. And there was quite a bit of seizure activity going on, about 28% of the time spent in seizure. Here's what happens in this bilateral prep. So something actually quite interesting happens here, and we're still trying to figure it out. So here's the prep here. This is left side and the right side. I don't know if you noticed, but there's a high amount of synchrony. I mean, very high. Here's a plot of the cross correlation between the two sides. And what you see is something very bizarre. The delay is about 0, even though it takes 6-7 milliseconds to cross. So a signal from left to right takes 6 milliseconds. Yet the delay between these two is 0. So they're perfectly synchronized. We've seen this in these slices, and it's not just this. We've seen it in vivo. There is a high degree of synchrony in these slices, even though my colleagues, including Mohammed, tells me that in patients you don't quite see synchronized seizures like this. But in these animals, this is very interesting because we need to explain how do you get 0 delay between the two sides. So that means that these two sides, which are connected, are seizing or spiking exactly at the same time. So we have a computer model now that reproduces this phenomenon. I won't talk about it because we don't have time, but you can see, though, if you cut this path between the two, you still have seizures on both sides, or seizure-like activity on both sides, but they are no longer synchronized. There's no cross-correlation. Cross-variance is high, and if you look at the timing, everything is spread out. So this is a very interesting phenomenon where you get seizures on both sides, and you can predict that what I'm going to tell you is that if you put an electrode right here, one should be able to stop seizures on both sides simultaneously with a single electrode. And here's what happens. So this is a plot of the proportion of time spent in seizure activity in this preparation. This is for a unilateral prep, and you can see before, during, almost 0, and afterwards it comes right back. This is for the bilateral prep. The bilateral prep is actually better at suppressing seizure for some reason. We don't quite understand that yet, but there's no seizure, none, 100% suppression in this preparation. This is pre and afterwards. So this is quite effective, and I will show you now some results of the mechanisms of this effect. How does it work? And we just submitted a paper on this, so you get to see some unprinted results yet. So how do we pick this 1 hertz, the frequency? I told you we wanted to use 1 hertz, but why not 4 hertz? So why not 10 hertz? Somewhere in that range. So we worked on this a little crazy idea that is controversial, that interictal activity is protective of seizures. Now that's very controversial. Some people agree, some people disagree. But we had experience in other preps suggesting that might be the case. And you can see that when you have seizure activity starting, you get spikes starting, and then you get seizures. So perhaps the tissue is trying to protect itself from seizures. You could argue either way. Anyway, we decided to base our stimulation on the frequency of interictal spiking activity in this prep. And so we did a Fourier analysis, and there's a spike here at 1 hertz, and we decided to pick that frequency of the this 1 hertz to figure out to do the suppression. Here's some example of this activity here, and here's actually intracellular recordings to show you what the cells are doing intracellular. This is a sharp recording from neurons in this preparation during a seizure. You can see this very large depolarization called paroxysmal depolarization shifts, PDS. They depolarize like crazy. They fire a lot of action potential and calm down a little bit and come back and keep going. Here's what one slide that summarizes the mechanisms. I won't spend too much time on it, but it's sort of interesting. First thing to notice is that this phenomenon is amplitude dependent. Here's the and frequency dependent. So you can see the effective frequency. 1 hertz was the best. 3 hertz. So we were right in picking 1 hertz, right? Because it turned out that 3 is not as good in this prep. Okay, that doesn't mean it's true in everywhere. 10 hertz was not nearly as good. but 1 Hz was quite active. And so then we did some pharmacology experiments to try to figure out what was going on. And we used basically 2 blockers because we focused on these by eliminating others. And we looked at other preps as well. So is this phenomenon robust? Where this is the suppression in 4-AP. So you can see 100% suppression right here. This is bacucolin, another epidemiogenic model. And you can see that there's also 100% suppression. This is yet another model of epilepsy called low magnesium. If you reduce magnesium, then the NMDA receptors are open all the time. Glutamate is activating them. You get massive seizures. And so in low magnesium, which is another model, yet this low frequency was still able to block this. But if you add sacofen, which is a GABA B blocker, then you see the suppression effect is down 40%. And if you add clotrimazole, which is a AHP is a long-lasting hyperpolarizing potential called after each action potential. That is a calcium-induced potassium conductance. You can see you further reduce this. We eliminated already the GABA B blockers. They don't work. You can see that anyway because bacucolin is a blocker of GABA A anyway. So these 2 potentials, these 2 blockers rather, were able to block this effect. And if you, I don't have the data to show you, but if you add both of them together, it's all gone. We cannot suppress this anymore. Now this is not too surprising because GABA B response is very long. It's like about a second long. And so is the AHP. The AHP is very long-lasting hyperpolarization, about also a second long. That means that these two blockers are activating channels that maintain, we thought, the cells in a hyperpolarized state. And so we did some analysis on that, and I'll show you some results. You can see here that is a further plot of the frequency. But what's very interesting is the effect on the distribution of the activity. So during the stimulation, activity is right at the beginning and then nothing afterwards. You can see this right here, all the spikes happening, and then after that, everything quiet. So what the stimulation does, it forces the cell to fire early, right at the stimulus, and then nothing after that. And that nothing is maintained by these 2 hyperpolarizing potentials. If you block these potentials, then the suppression effect goes away. And then you can see what's happening in more detail here, because if you look at the resting potential of these neurons, this is before stimulation, the cells are depolarized and fire. This is during LFS. You can see the resting potential is down. So here's the resting potential here is down. You turn it off and it's back up again. This is measured right here. So what the stimulation does, it enhances these long-lasting potentials. These cells are hyperpolarized during every pulse and maintain during the duration of the stimulation. Now, so we're still working on this and trying to publish this, but we were interested in to see whether, this is very nice in vitro, but does that mean anything? Most of the stuff we do in vitro, it's hardly applicable to humans. So first step is, well, can we do an in vivo preparation? Does that work in vivo? So we came up with a preparation that was published actually just last year, a few months ago last year. And so this is actually quite powerful because the model that we use is more of a status epilepticus model where we inject 4-AP in an in vivo preparation, acute in vivo. And these animals are injected every hour and maintain a continuous state of epilepsy. So we call that status in the rat model. But there's continuous seizing activity for hours. Then every hour you turn on the stimulation, turn it off, turn it on again, turn it off, turn it on, left and right. You can see again the synchrony right on both sides. And you can see that even in this model, which is continuous seizures, this low frequency stimulation can clearly suppress seizure activity. And that was quite surprising to us because that is very nasty sort of seizure activity in these rats. And we did not think that this would work, but it's quite robust. The next step, of course, is does that work in a chronic model of epilepsy? So there are many different models. We use a model from Nissinen, which was published in about 2000. In this model, the reason I chose it is because it does not require any drugs, and it does not require kindling per se. So what happens in this model, you take a rat and you inject stimulation in the amygdala once for about 20 minutes. The animal generates a status like seizure, fairly long seizure. You turn off the stimulation, and then you let the animal go back to his business in his cage. And about a month, 2 months later, it takes a while, it starts seizing simple seizures like this one, but a few seconds long, and not that many either, maybe 5, 10 a day, something like that. So it's a fairly sort of mild model, and the reason I liked it is because when the animal is generating the seizures, there are no drugs on board. It's not generated by drugs. And in the study that we published a while ago on this model, we did not see any damage much to the hippocampus. So to that respect, it's not a great model, right? Because clearly there is damage to the hippocampus during seizures. Here's our model of, definition of what we call seizure when we do not call seizures for detecting. So this is a LabVIEW, custom-made LabVIEW software that we developed. And I say we, students and postdocs. And so basically, we have to differentiate between movement artifacts and seizures. There's quite a few movement artifacts. And you can see, for example, this is the seizure, but this is not. And the reason is, the criterion that we use is 7-second long event, and it has to have frequency. This is a Fourier analysis of the event at the same time that we're looking at them. And you have to have frequency in the past 30 hertz. Whereas a movement artifact has also very long lasting, but there's no high frequency. Here's the two events. It's hard to say which is seizure, which one is not. But this is with this criteria, I think we're pretty confident that we're detecting. So we did a study that was also published recently in rats, in this model, 7 rats. Every single rat had a seizure reduced. Average reduction, here's a plot of that. So the point of that seizure is that there's 100% responders here. Every rat was suppressed. Here's a plot of the average is 90% reduction in this model. And that came out in Epilepsia last year, 2 years ago, I can't remember. There is a residual seizure reduction that happens. So this is 2 weeks before, 2 weeks stimulation, 2 weeks afterwards. And that we do not quite understand how that works. There's maybe some changes in the network that produce this response, but we don't know. So now, we were very encouraged with that idea, of course, in rats. Let's go see a neurologist or neurosurgeon, see if we can translate that into humans. So the first thing we did is say, well, what is the equivalent of this track, which is in a rat, ventral hippocampal commissure, in humans? And so we did some searching and did not find anything significant in humans, so we're sort of disappointed. But there is in humans a smaller tract called the DHC, dorsal hippocampal commissure, which is highly unknown. There were at that time only 3 papers on this track, and it's sitting right here. You can see it right there. This is a fimbriae fornix hippocampus here. The fornix is here. This track connects these 2 fornices here. It's sitting right underneath the splenium of the corpus callosum. I think I have a picture later on to show you. It's pretty thin, and no wonder it's been sort of ignored because it's not quite prominent, quite a prominent tract. So anyway, the surgeon knew about this track and said, yeah, we're doing experiments, we're placing a lot of electrodes for our patients. We could place one more, so this is number 11, here, dorsal hippocampal commissure right here. So he's sitting right there, you can see this track right there, right underneath the corpus callosum. Very thin track, not heavily myelinated either. And so while the patients were in the epilepsy monitoring unit, the electrode was placed there, and Dr. Kubisi, who's a neurologist in charge at the time, was running studies at about 1 hertz anyway to do monitoring, to see, if I stimulate this electrode, say here, what do I get here everywhere, which is a really interesting study. I was much more interested to know what happens if you stimulate 11 and do we suppress seizures. So we started slowly, but we did a few patients, and here's an example of what happens when you stimulate this track. In this particular patient, one electrode was able to activate evoked responses bilaterally along the hippocampus, dorsal middle temporal, on both sides, or in some cases it's not quite as prominent, and it's not always as good in all the patients, but clearly it's possible to activate the whole hippocampus, just like in a rat, with one single electrode, and bilaterally. So in principle, the idea that we developed with this rat might work there. So we have done a preliminary study with 11 patients, and the results are not published yet, but we're working on that. We were getting, we did stimulation of about 4 hours long. Again, this was run by Dr. Kubesi. And we didn't have much time to stimulate because this is not a real clinical trial. Patients were in the monitoring unit just for short times. So while the patient was lying, we did this study where we would stimulate for long, 4 hours periods of time, sometimes 2 chunks of 4 hours, and look for the probability of getting seizures. And according to the statistics, there is an 87% reduction in seizure in these patients. So this is by far, if it's true, right, the largest, the best suppression mechanism that that's been reported. We did a few simple tests, memory tests, because this track might involve memory. So the question is, does it affect the cognitive functions? And there was no complication that we could see. This just to tell you a bit more about this tract. The DHC, there's a few recent publications. This is a publication from a group in Montreal. And they did diffusion tensor imaging, and in 20 patients found this tract. And all patients' tract is definitely there. You can see it's in the green fibers here, the cross, and here the crossing again. So it's definitely there. And so what's happening with this now is we're in the process of trying to expand that study to a real clinical study where there's a device implanted in this particular area. Actually, we'll probably have 2 electrodes a little bit closer to the fornix, and still hitting those DHC targets. And we'll see if we can replicate that in a real study. So that's where we are with that idea. So now let me show you the next frequency range. That's high frequency. Now this is a frequency range that is mostly used because that's what DBS uses. 130 hertz, 150 hertz in that range. And everybody is using that. And strangely enough, very few people are using low frequency. I'm not quite sure why that is. But that frequency is used in epilepsy quite a bit now because of the success of high frequency to control motor disorders and Parkinson's disease. So we, as it turns out, have done some work in there too, trying to understand how do neurons respond to this kind of frequency. What happens to a cell when it's exposed to this high frequency? Some of the targets that have been used for epilepsy is central median thalamus, STN, of course, hippocampus, This is anterior nucleus of the thalamus, which is the target of the last big trial paid by Medtronic, called the Santé trial, which had some positive results but never got FDA approved. They do have success in Europe, and they are using this technique in Europe. So they use high-frequency stimulation of this target to produce reduction. So what's happening with high-frequency stimulation in DBS? That is still unknown. There's a whole slew of possible mechanisms. Nobody's figured out exactly how DBS controls SDN. I just showed you this example here. The synaptic depression is a possibility. Informational legion is another possibility. Depolarization block is a possibility, etc. So what happens for epilepsy? There's basically two big groups. This is Medtronic, and this is a Santé trial I told you about. Here's some of the results: 40% reduction, 14% became seizure-free. Neuropace is another company. They use a whole different technique. They use recording and stimulation at the same time, and they try to record activity to detect the focus and stimulate, and they're getting some very good results, but not all that different from this. And they are trying to get FDA approval, and they're running into difficulties there too. Now, what does happen to neurons when you expose them to that kind of frequency? So here's an example of, this is work we did quite a long time ago, in 2001. Here's a model of epilepsy. This is in vitro again, where we do low calcium. You can see this activity. You put the stimulus, electric field at 50 hertz. And at 50 millivolts per millimeter, nothing much happens. But if you turn up the fields a little bit, the 160 millivolts, then you get complete suppression. If you use another model, high K model, high potassium model, same thing. Picotoxin, which is a GABA A blocker, also a good model, you can block it. So this is very robust. So you can put a high frequency, and you can block synaptic activity. So to show you more about what's happening, we did some intracellular recordings, look at cells. Since you're passing a large amount of stimulation on the outside, you need still to know what's going on to the transmembrane voltage, because that's what turns on the sodium channels. So what we have to do is to record inside and to record outside of the cell, take the subtraction, so VE minus VI. And here's what you see. This is a transmembrane potential, and once you remove the artifact, you see this. The cell is firing in a very potential. The reason it's so small is because we have a low-pass filter there, but it stays depolarized. So this is a typical example of what's called depolarization block, where the cell is overexcited, depolarizes, because the membrane voltage is high, then the sodium channels are blocked and can't fire anymore. To turn off the stimulation, it goes right back down. So that's an example. What possibly happens, we don't know if it happens in every cell, but it did happen in that cell. The other thing that happens is, around these electrodes, is in our hands a very high increase in potassium concentration. Potassium increases, and you can see 100 millivolt per millimeter goes from another 1.5 millimolar of potassium increases. Here about almost 2 millimolar potassium increase. And this is concomitant with the suppression. So we think that there is a relationship between the potassium concentration increase and the suppression of the activity. The link has not been causally proved, but it's pretty strong. Then there was some argument about, well, maybe the cells are responding that way. What about axons? Because at that point, there were some issues about do axons respond differently to cells? Maybe axons are responsible for the suppression during DBS, or maybe they're excited or inhibited. We didn't know. So we decided to do a study just on axons exposed to high frequency. So here's an experiment that shows a plot of the CAP. The CAP is a compound action potential, which is purely axonal. So this is all the axons working together at the same time, generating a voltage. That can be measured when you excite them. And you can see right there. So here's a stimulus artifact. Here's the compound action potential. Small, very small, 0.2 millivolt here, right? But measurable. And this is purely axonal. But during high-frequency stimulation, it's completely gone. You turn off, stimulation is back up again. And you can see the suppression here as a function of the current, 250 microamp of current, which is sort of standard. It's clearly sub-threshold, though, right? But what's interesting here is the frequency plot. You can see that maximum or optimum suppression happens to be right in the range of 130 hertz, 150 hertz. And then after that, it's no longer effective for reasons we don't understand. And it's not so effective down here for reasons we don't understand either. But the optimum frequency is in that range. How is this suppression taking place? So here's an example to show it's not synaptic, because we can repeat this experiment in low calcium, no synaptic activity. And you can see that here's the synaptic signal right there. You block it with low calcium, and you get still a nice compound action potential. You stimulate, it's gone, and it comes back. So this CAP clearly disappears, and it's not a synaptic phenomenon. Could it be desynchronization? So all the axons no longer fire at the same time, they're still firing. We don't think so. In fact, we have some new data on this, which I don't have here, but if it was desynchronization, we would expect the compound action potential to spread out in time, be delayed. None of that happens. We do have some intracellular data that shows it's not that, because we don't see any incoming action potentials coming while recording from the neurons, so we don't think it's that. So to isolate the problem even further, to be sure, what we did is did a special axonal preparation. So this is a different preparation, in vitro preparation, where we isolated an axonal pathway. So here's this, dark here is the alveolus, which is the axons of the CA1 neurons. Cut that off, and here it is right there. It's by itself in a dish, exposed. And we did experiment just on that to eliminate every other possibility. And same thing happens. You can block it with high frequency stimulation. Clearly, axons cannot follow this frequency. And that was somewhat controversial and still is. People do not understand. Other scientists have argued with me on, well, I know that axons can follow at this frequency. Why is it that they cannot follow here? We know that cells can fire high frequency. What's the problem here? Well, one problem, of course, is they follow if they stimulate it from the inside. But here, this is external stimulation. So when you stimulate externally, you activate a big chunk of tissue. You change the extracellular space. Everything is different from before. We did experiments where you can put a cell and stimulate at that frequency, and it follows fine. But if you do it externally, that's a whole different ballgame. Then we repeated some of the potassium measurements that we did with axons as well, and clearly same thing happens. Potassium is going up, highly correlated. This is potassium concentration and the amount of suppression. And we also did an experiment where you apply potassium concentration and you can see suppression. You can cause suppression in high concentration. So clearly potassium is involved in this. This is an experiment to show it's happening in vivo as well. And so at high frequency, this is 100 hertz. We stimulate this tract that you're familiar with now, the ventrally hippocampal commissure, in vivo, and record from the CA3 on both sides. Here's what happens at very low frequency. Get a nice evoked potential left and right, both sides. But when you turn on the stimulus at much higher frequency, you can see this very slowly disappear. There's nothing left. And then eventually it comes back afterwards. So again, in this situation, these cannot follow. Here's a plot of this normalized CA3 amplitude, so the evoked potentials are dropping. and this is during high-frequency stimulation, all the way down sometimes to 0, and then come back afterwards. And here's the frequency ranges here. And then if you plot this maximum here at this frequency, what you see again, the optimum frequency is around 150 hertz, which is sort of surprising. Interestingly enough, there's also some effects on units. So if the cells which are normally firing at this high frequency rate here, this is unit activity from CA3 cells on the left side and the right side. There's a stimulation here, and right afterwards, the units stopped, and then eventually they can come back. So the effect of the stimulation is also on the units. Here's the, you see them coming back here, back to normal, but right after the stimulation, something's happening to the cells. They no longer want to fire. And this has been quantified here, and you can see that just 5 pulses is enough to stop units. 10 pulses, 100 pulses, 3,000 pulses, there's a long it takes a long time for the units to recover. We haven't published yet, but we're working on that. So I'm going to hit the conclusions. And so I hope that I sort of showed you the electrical stimulation, which is normally an excitatory event, can produce suppression. And talked about the DC. DC currents can hyperpolarize tissue and prevent and decrease excitability. The mechanism is known. It's membrane polarization. But it's very difficult to use it because it causes damage and it requires very specific orientation. Low frequency, I wrote this slide a long time ago. Now we do know what we think we know what the mechanism is. And it has to do with the long-lasting hyperpolarizing potentials. And then the high frequency can reduce neural excitability. That mechanism is not clearly identified. I mean, we think it has to do with depolarization block and potassium release in the tissue, but that has not been proven yet. So these 3 mechanisms, or the 3 modes, can produce seizure reduction, and some progress has been made in the determination of the mechanism. So here's some of the people that work with this. Of course, you know Dr. Kvesi here, but Marianne Worth, also at the clinic, was working on this with us early on. And Dr. Luders has been involved. These are the surgeons. Dr. Masunos, who unfortunately passed away recently, was the first one who started to work with us. And then Dr. Miller is now working with us. So we also did some work with Dr. Najim at the Cleveland Clinic. And this is a colleague from Zhejiang University in China. She's working on a paper on high-frequency stimulation, which actually shows that even if you suppress an axon, the cell body itself, I don't have the data to show you, but the cell body itself is still excitable. So it's a very localized effect. And the neurons themselves are not affected. You can activate them from a different path. So we're still obviously working on this. And then there's a lot of people that work with us on these projects. Dr. Rashid did all the work on the in vivo chronic preparation, and we have an animal technicians very active. We have undergraduate students also working on this, and a lot of graduate students and colleagues have been helping us with this. And I'd like to thank you for your attention. Be glad to answer any questions that you may have. Yeah.
Can you comment on how well? So, can you comment on how well the animal models predict ability to translate therapeutic interventions to humans?
Right. I can try to comment on that. It's not an easy question. My experience with some of the work I've done and some of the work my colleagues have done is usually doesn't translate well. So that's why I'm sort of surprised that this particular project translated so well. I'm very surprised. And maybe it's because the mechanisms that are involved are general throughout animal kingdom, and so if they are the same mechanisms everywhere, then it doesn't matter when you translate it. So if all brains have GABA B receptors and SHAP after hyperpolarization mechanisms, then if you can activate them in a rat or a human, it shouldn't matter too much. But I've had many other techniques that did not work, could not be translated. A good example is the DC idea. I started working on that, you saw, like a long time ago, and I did not go into translation because I didn't think it would work. I mean, it may have worked, but it would have clinically it couldn't. So I did talk to Ben Abid, who you might know, in Grenoble, who started the DBS a long time ago. I said, can we put an electrode in the hippocampus and pass this DC current? I know it's going to block seizures. He said, well, maybe we can pass a wire all the way into the hippocampus and apply this current, but I knew there's going to be some damage, and I knew there's no way on earth you can put it exactly where it needs to be, and even if it was, the orientation of the neurons are not the same. So I just gave up on that. And there are many other ideas I gave up on. I didn't think they would work. A good one is something I didn't say anything at all, is this resetting idea. I worked on that for quite a long time and published a couple papers, not much, and basically trying to copy what's happening in the cardiac area, where you take one pulse and there's a vulnerable window. If you hit it just right, you induce fibrillation in the heart. I said, well, if we do this in the brain, then we would desynchronize all these neurons. It'd be perfect to prevent seizures. And I found the model is one high potassium model that has very regular periodic activity. You put a pulse there, you could maybe sometimes hit it, but it was impossible to find.
I have a question.
Yes.
The current understanding of the mechanism of action of low-frequency stimulation with prolongation of the hyperpolarization and what you mentioned, does it explain different actions of low-frequency stimulation on the hippocampus itself compared to stimulating a tract that is diffusely connected to the hippocampus?
The mechanism that you saw, that we investigated, are actually tested in the bilateral preparation, meaning that the recalling from cells and the excitation that you get from the 1 Hertz is coming from these tracts. So these are not a different way of studying it. We're actually stimulating that VHC in rat and producing this long hyperpolarization. So what maybe it did not come out clear is that what's happening is this low frequency stimulation neurons normally tend to fire any time. They just keep on firing. But when you apply low frequency stimulation, you force them to fire once at every pulse, and they don't fire in between. It takes a while to prevent seizures. It doesn't happen right away. It takes maybe a few minutes, 5 minutes, and then slowly the seizure will stop. Then the stimulation maintains that the tissue in a state of just slightly hyperpolarization because of this long-lasting potential, and then seizures just don't want to propagate in there. And they don't generate seizures themselves. So the mechanism that we observe are those generated by the axonal stimulation, because we're using that same prep.
Thank you very much.
Thank you.
You probably haven't met Dr. Kaminsky when he was...
Hi.